Bring it.
Dropping it. Okay, great. Thank you all for coming today. So today is our first seminar of the fall. Meet a data scientist. So this is one of the series that we started last year as part of the Institute for Data Science and Computing in order to introduce data science in general. And also this is actually what we wanted to be a little bit of a different type of seminar than the normal academic seminars that you go. It's more about, we select the speakers to tell us a little bit about their experiences, what they are doing, how they ended up coming into this space, some of the grand challenges that they are facing. It's not really only how great, the type of work that we do is, but also what is the potential of the work that we do, and what are some of the perhaps issues that one faces in this. So it's basically, we ask the people to describe a little bit more about the journey that they take in order to come into the data science, since data science is not technically a discipline that it's been taught at the universities at this stage. We hope that this is going to change, and this is part of our mission as of Institute for Data Science and Computing, is to actually see some of those changes. So today, we have a very special guest to kick us off for the fall semester. We have with us Athena Hadjixenofontos. Dr. Hadjixenofontos, I should say, is actually, many of you know, she's an insider. So she's been, at the University of Miami for a while at different roles. She was also part of IDSC at its previous incarnation. So Athena got her undergraduate degree in biochemistry and genetics from University of Leicester in UK. And then she came to US and she did her PhD here in Miami at the Department of Genetics and the Institute for Human Genomics in our medical school. And then after that, she did a postdoc at Yale. And after that, she joined actually IDSC, or the Center for Computational Science at that point. And now she actually is a vice president for a major financial institution that we are not naming. So because I think that the Athena is one of the, how can I say, illustrations of how data science evolves, how data science it moves from someone like a background from Athena that had got a very biological, biochemical, genetics background to something that it actually applies even in the as far as away as in the financial industry. So with that, and without no further ado, the floor is yours, Athena.
Thank you, Nick. Thank you for the kind words and for the invitation. And like Nick said, I'm a proud UM alumna, and I'm just so happy to be seeing some familiar faces today. Thank you for taking the time out of your day to attend. And I hope that what I have to share is worth that time. So my area, my sort of special type of data that I work with throughout my journey, it has been high dimensional data. And that's most of what we'll talk about. So we'll start, oh, excuse me, I think I'm, okay. Trying to put the Zoom panel meeting somewhere where it makes sense. Okay. So first, obligatory disclaimer that all views and opinions expressed in this talk, anything that I say, is solely my own and do not reflect any views or opinions of my employer. And here's a high-level view of what we'll cover. So a little bit about me, as Nick said, who gets to be called the data scientist these days, and I'll add a few of my own views there as well. And then we'll do a very high-level survey of what are high-dimensional data and how we work with them. How is that different than in other, when you're working with much lower dimensional data. And again, this is geared towards a sort of budding audience, so maybe more lay people who seek to peek into this world. So if you're an expert, most of what I say you'll probably be very familiar with. Alrighty, so this is me. I was born in Cyprus, so my geopolitical journey starts there and raised there. My dad is an engineer, and we were just a science family from the get-go. So I think that's really the first seed of how I ended up in data science. I moved to the UK for university. I thought I was going to do marine biology. I ended up being really interested in biochemistry and genetics. And I lived in Portugal for a year, where I did research in microbiology. So I have a lot of wet lab experience, more than necessarily a data scientist needs. And then I moved back to the UK, where I worked in cancer for a while before coming to Miami. And it was in my PhD where I kind of changed direction, right? So at university, I studied biochemistry and genetics, and I was lucky enough to graduate top of my class, and I was super into yeast genetics. And I got to work with a Nobel laureate, so with Paul Nurse, who worked on the cell cycle. And I thought I was set on a path of the classical genetics. But then I got to my PhD rotations, and this is where you try out different labs for a few months before committing to one of them for the next 5 years of the program. And in my first rotation, I started feeling really bothered by how isolated my thinking was, right? So for every single one of my projects prior to that point, I was focused on one thing, right? On one gene, one protein, one at most pathway. And that just felt really limiting, right? I started wanting to see biology from more of the whole system perspective. And this is really what landed me in genomics. Genomics was the first omics field, right, back before all the other omics became possible. And in genomics, I found other things to bother me, right, other than, and we'll talk about some of that today. And then let's move on to sort of how do you go from genomics to NLP? So my first NLP project was at the previous incarnation of IDSC, working with Nick and some of the just amazing people at So we had a project, my first NLP project, and I discovered that NLP has a lot of the same methodological issues that genomics does, somewhat also because of the high dimensionality of the data. And NLP has a lot of different sort of types of subfields and subtasks. So I'm sort of focused on the type of task that I was doing in genomics as well, right? So building predictive models and mostly classification type tasks. So that was the rationale for that transition. It felt natural, and we'll see a little bit in more detail what those similarities are later on. And also language data, right? Language data, they're complex, they're messy, they're ambiguous, there's redundancy. It's like humans don't speak in very clear terms, right? So, and if you ask any biologist, biological systems also have those same characteristics. So the NLP transition felt very natural. And when our first child was born, so I reluctantly had to leave Miami so we could live closer to my wife's family in Phoenix. And I even more reluctantly left IDSC and joined industry. So if you have any questions there, we can also talk about sort of high-level view of contrast between working in academia and working in industry. I am enjoying working in industry. It's definitely a very different focus, very fast-paced, and yeah, you also still have a lot of the sort of interdepartmental, in this case, right, communications that can really make your work a lot easier. So most of the soft skills really translate, right? Learning to work in cross-functional teams and with people who don't speak your language. So that's the other piece that informed what I put together for today, which is, again, you know, so that anybody can understand it, right? From the practice working in cross-functional teams. Okay. All right, so that's a little bit about journey and sectors and domains. So let's jump into high-dimensional data. Actually, Nick and Janina, do you want to do questions sort of as we go, or do we leave time at the end? How?
It's up to you, Athena. So,
okay.
You know, and the audience, if they want to ask a question, you can actually put it on the chat and then we can alert you, or if somebody wants to have a question, you can raise your hand and you can ask a question. So we want to be flexible about that. So we want to be a little bit more informal, but it's up to you, Athena, what you prefer.
I like questions as we go. I think it's easier when it's embedded rather than, you know, out of context at the end. So if you have a question, if you please put it in the chat. And Janina, I don't have enough screens to be seeing the chat at the same time. So if you can let me know.
No worries. Yes, we'll let you know.
Okay. All righty. So shifting gears from sort of my journey to talking about just some background about working with high-dimensional data. So what is a dimension, right? Let's start at the very beginning. I get this question from people sometimes, so it may sound basic, but I think it's a good place to start. So in our context, it's another name for a variable. It's a column in your spreadsheet. It's an attribute of an observation. It could be of any kind, you know, those are listed there. And then when you start using that variable as an input into a model, then you're starting to think about it as a feature. And that may or may not be the original, you know, untransformed dimension, or it may be something that was derived from it, or some combination of dimensions into a single feature, and we'll see more of that later. So that's not, you know, those two things are not always the same. And here's a little tiny toy example. So we have some people attributes here, and there's a huge, right, number of people attributes. If you think about, you know, the number of cells in your body, and each of those expresses how many different genes at different times in development, right? The number can become really huge of what is possible to collect. And some of these things are going to be relevant, right? And some of these things are not going to be relevant to what you care about. So if you do care about gene expression for a specific disease, that's relevant information. If you care to see, you know, do people who, I don't know, what may be more sort of psychology phenotypes or other things about people, then you're going to be interested in a different subset of those possible variables. So here, I think one of the points that I wanted to make is how subjective it can be to decide what it is that you're collecting data on. And so I have here favorite animal at age 3. Is that important? Is that predictive of anything? Maybe. I have a 3-year-old, so it's kind of, many of my examples I think you'll see are, are informed by those worlds. Okay, so if we think about the sort of space of possibilities, right? there's just and seeing everything as data, then all that data is high dimensional, but you end up limiting what you actually collect information on. And that can end up limiting the dimensionality of your data set. So, and that's generally a function of availability of resources, or if you have some domain knowledge that tells you what you want to focus on, so that you can choose variables that are suited to the task at hand. And this is actually one caveat, I think, in the era of big data, because a lot of the automated collection generates gigantic data sets in terms of how many rows they have, but what you're collecting data on is generally more limited. And even with big data in terms of rows, right, the data set isn't necessarily then going to be collected with the objective of your task in mind. So there is always this awareness that you need to have about, is it suitable for your task at hand, or is the definition, right, of what maybe they used not relevant here, okay? So let's start moving on a little faster, I think, just doing a quick time check there. So I've mentioned some of the biases, right? Think of that, the elephant and the man who are sort of trying to feel their way around. Like, you're going to bring that into your analysis, guaranteed. So the best that you can do is to be aware that you're doing that, and try to find areas where that's actually impacting what your output is. Okay. So how high dimensional is high dimensional? So for those who work with structured data, thinking about like 20 dimensions sounds like a lot. It's a lot when you're thinking about visualizing it, but it's not really right in the range or in the order of magnitude to create some of the more serious issues. So when we get to 10,000 variables, when we get to 88 million variables, it becomes a huge issue, right? How do you work with that? So right in statistics, you'll see P and N. P is generally what we use to denote the number of parameters. So these are the number of variables you're going to estimate these parameters for whatever it is that you're building. And N is the number of observations. So when P is much, much larger than N, a lot of your sort of classic statistics assumptions start breaking down. So let's see what that looks like. So why is it important, right, that you have such a small ratio of data points to parameters? There's a number of reasons for this, and it's collectively referred to as the curse of dimensionality. This expression is attributed to a computer scientist, Richard Feldman, who was working in dynamic programming. But really the heart of what we're talking about here is that when you have this many dimensions, the data ends up being very sparse. And all of your asymptotic theory, all of your statistics is no longer in your corner, right? So if you're thinking about it in terms of, usually we try to find similarities, right, between data points, to try to find some structure, something that can tell us something. When the number of dimensions is so large, there's lots of differences between the data points, and it becomes more difficult to figure out which of them matters. And if you think about it, it's sort of more, yeah.
No, finish your thought. We have a question, but finish your thought.
Okay. So if you think of it sort of in 3D space, right, where we can visualize this, sort of think about the shadow, right? So say there is a tree behind you, and there's the shadow of the tree falling in front of you, and you might see like a bunch of apples, right, that in the shadow of the tree look like they're all together. But then when you actually look at the real tree in 3D, right, go from the 2D shadow projection to the 3D, the apples end up being much farther apart, right? So now we're just going from 2D to 3D, right? So go to 88 million, right, dimensional space, and the sparsity issue is a real problem. Yeah, Janina?
So the question,
so we have a
sorry.
question. Oh, sorry. I was gonna
No,
say,
it's
Pedro,
okay.
go ahead and ask your question.
I was wondering if there's any tools or applications that you use to visualize some of these higher dimensional, or is that just not done? Is it more like a mental?
We'll see. Hold on to that thought. Most of the rest of the talk is about that. Not just about visualization, also about modeling with it, but visualization is a big piece of it for sure. Okay, so yeah, let's move on. I think the point is made, right? If you have any questions about that, we can talk more about it. And what does that look like in real life? So let's start with the example of genomic data. This plot is called the Manhattan plot. If you squint your eyes, you may see something that looks like skyscrapers, right? So therefore the name. And each dot on this plot is a position in the genome that varies between people. Nowadays, we usually call it, variant. Variant is the catch-all name for those. And you have data, you have collected data on those. And the objective here is to see whether any of these variants are associated with whatever disease you're interested in, right? So this is the design of a classic genome-wide association analysis. And the data collection happens by the machine, right? By the genotyping machine. So you have all variants collected in the same experiment, say with a genome-wide chip back in the day, and you end up with 1 million variables for each of the people that you genotyped. So if a typical data set, right, from a single lab is a few thousand people, when you divide your N by your P, right, so 1,000 people and 1 million parameters, you end up with that ratio in the range of like 0.001, which is much, much less than the 10 to 5 that is recommended for working with sort of traditional statistical methods. So this is why you see labs coming together as consortia, right? So everybody pulls together all of their samples so that they can actually say something about this data. And so calling something important out here, although this is a high dimensional data set, right, from a sort of visualization perspective, the models that were usually built for these genome-wide studies are not actually considering all variables in the same model, right? They're first filter out a bunch of them. So for various reasons, maybe they're missing too much data or they're correlated with each other. And then we take each of them separately, build it its own model. So you end up, instead of one model with 1 million variables, right, which we've established is a bad thing to do, we end up with 1 million models, right? The cost of this is that you then have to account for the fact that when you run 1 million tests, you're going to, by chance, find something that looks important but actually isn't. So you have to adjust your thresholds for accepting something as important, as a significant result. Okay, let's move on to what natural language data look like in my adoptive world of NLP. So natural language just means that it's languages that are spoken by humans. So English, Greek, Spanish, right? All of those are natural languages. And the data set here is a corpus. You have a collection of documents. They could be emails or books or instant messages or tweets or legal contracts, any sort of collection of documents that you can imagine. And what is the dimensionality of that corpus? So the way that we represent, I have a tiny little corpus here made of 2 documents. So pandas are my favorite animal, and my favorite dinosaur is Mosasaurus. That's my son's favorite dinosaur. It's a very cool dinosaur. And the way that we represent that as, in a form that the computer can understand is by building vectors that are the size of the vocabulary of the corpus. So with my little toy corpus, right, I took the unique words and put them in this table. And for each document, right, for document 1, I now have ones in the places where the document has that word in it and zeros in the places where it doesn't. So I end up with these 2 7-dimensional vectors that would be my input, right? So it's now encoded in the form that the machine can do something with it. This is called one-hot encoding, and it's actually not a super great way to represent it, but it's really simple to explain. It's not super great because it doesn't preserve a lot of information, like the order of words in the document is lost, or whether you have maybe 2 words that occur together, like New York, right? You would have a 1 for the word new and a 1 for the word York, but that's not really what that means. There are, you can build bigrams like 2-word pairs and start increasing the dimensionality even further by adding all combinations of 2 words, right, at a time. Yeah. There are more sophisticated encodings, and if we have time, we can talk about them. So what is the dimensionality of the corpus? Yeah.
Question from Mustafa.
Yes, what is the question?
Do you want me to read it, Mustafa, or you? Okay, I'll read it. Hi, what about the data situation with columns? Can we still apply the tools that you've used for visualization? For example, how can we plot a scatter matrix? Also for visualization and dimension reduction, do you rely on unsupervised techniques?
Yeah, we'll talk a little bit more about dimension reduction later, and there are some supervised techniques that you can use too, and actually that's a pretty hot area of research right now. There's a lot of papers coming out for supervised dimensionality reduction, which is very cool. In terms of plotting, you're definitely more limited. We'll see a couple of examples in the exploratory data analysis part. So yeah, that's the next section. So hold on tight. Okay, so what I'm trying to say with this slide is, what, again, back to that question, like what is your sort of order of magnitude of dimensionality for natural language data? So an upper bound, right, with 170,000 words in the English language, that's about, as big as that would get if you only take single words, right? If you only take unigrams and you don't start combining words into separate variables, right, essentially. And this graph shows the relationship between vocabulary size and the number of documents in the corpus. So there is a prediction of how many words you're going to end up with just based on how many documents you have. The dashed line is the prediction based on Heap's law, and the solid line is an empirical estimate based on a corpus from Reuters. So this is Newswire stories from 96 and 97. There's about 800,000 documents in it. And for the first million or so tokens, so tokens would be the individual sort of words when you break up the sentence. For the first million of those in this Reuters corpus, the prediction is around 38,000 terms, and the actual number is just a few, tens of terms off. So typically, like, you might end up with, say, 10,000, right? A vocabulary of 10,000 words is fairly typical. Okay, so now to Mustafa's question about how do you visualize this stuff? So just a couple words on the purpose of EDA. Why do you even want to visualize it and get to exploring it? EDA is not just visualization, right? So you might even build like an actual model to explore. We'll see some of that later. but a lot of it is visualization. You want to do it to identify, are there subsets of the data that you can't trust, right? Did something go wrong last Tuesday when we were genotyping batch 5, and most of that data is looking funky, right? So you do get a sense of that, of what you can trust and what you cannot trust. You also get a sense of the overall structure in the data. And by structure, we just mean are the characteristics of the data that you're examining sort of distributed randomly, or is there some structure to them? So if all of last Tuesday's batch was bad, right, there's some structure in the missingness of that data set, and that's important. You also build intuition, right? So some of your modeling decisions, I think it is really important to understand that you can build a model that is terrible and just misleads you, right? The fact that it produces an output doesn't make it reliable. Doesn't make it an output you can count on. So how do you know that you can actually rely on what you've built? Much of that will end up building on the intuition you've gained from doing EDA. So summary plots you can still do, right? So your usual univariate and bivariate descriptive statistics are good, except they're not for your individual variable, right? They might be for some aggregate measure. So maybe you have a mean and you plot the distribution of those means. So each data point that goes into plotting this summary plot has its own, other distribution underneath it. And you're just looking at this one aspect of that distribution. So of course, it's not giving you the full extent of the information that you may be used to getting from that. Yeah. Then there's also surplus plots. I'm a big fan of these. I think they're really great. It's more of a big picture view that they give you. And these are very common in biomedicine, but of course they can be used to visualize any other data. So here we have migration flows. So each color on the outer track is a continent or a subcontinent, and the form 2 information is shown by the distance of the bands from these bands that go across the circle from the outer track. So from Latin America to Europe is this big yellow. I know my colors. It's this big yellow one. So immediately, right, you can see that the largest volumes are from Latin America to North America, from South Asia to West Asia. And in the same plot, you get to see that Europe seems to be receiving the most diversity, with lots of little bands coming in from every other place. But people within Europe tend to migrate to other European countries rather than outside of Europe, right? So you see this green, the vast majority of that goes within Europe, but it's receiving a lot of these other colors. And it's a lot of information that you can encode in like a single graphic, right? It's really rich in information. And this one is even a simple one with more complex data sets. I've seen ones that have 3 or 4 or even more tracks, and each of those has information on a bunch of variables. So just huge amounts of information that you can encode in these. All right, now, actually, Mustafa, does that answer your question? Is that kind of along the lines of what you were thinking? You can type in the chat. Yeah.
Yeah, he has like tech audio issues.
Okay. All right.
He's asking, are these colors for clustering purposes?
So this is not your typical sort of unsupervised clustering type of plot. It's the color here shows just the continent that you're coming from. So you see North America is red, and then the people who are leaving North America to migrate to other countries are in this red band that mostly goes to Europe. with, you know, smaller volumes going to other places too. So it's not, yeah, we'll see some of those other plots later where it's labeled on top of what you did with the data. I think that's what you're getting at. Okay, so let's talk about dimensionality reduction. Already mentioned, it's one way to turn, you know, to turn your problem from a large P small n1 into a smaller p. And is the n large now or not really, right? Like you're not actually changing the number of observations you have, but it does change the ratio of n and p. So this sounds great, right? Except that the lower dimensions, with these methods, they're generally sort of squishing, right, a number of the original variables together according to how the algorithm decides to do that. And for some methods, it's a linear combination of the original variables, and for others, it's not. And the issue with doing that is that it reduces interpretability. So if you were to feed these lower dimensions, right, these combinations into a model and say, hey, model, how important is this lower dimension one? That doesn't even have an actual name, in discriminating between people with multiple sclerosis and those without, you'll end up with an answer, but can you use that, right? What does that mean? Yeah, we'll see how with genomics, you do end up with dimensions that capture ancestry. So it's something that's meaningful, that's interpretable, and that we do use, but that's not going to be the case across in every type of data set. There's also just quick note on supervised dimensionality reduction, and these methods try to direct the lower dimensions towards those that capture information relevant to the supervision target. So if your supervision target is the person, you know, that has multiple sclerosis or doesn't have multiple sclerosis, right? That's your supervision target. The lower dimensions that you are asking the method to capture are ones that contain more of that information. Yeah. I think I have a few more points to make here, but let's move on for the sake of time. So there's a lot of methods that you can choose from. PCA is the most common one that everybody will have heard. NMF is a little bit less commonly used, but it can be useful. TSNE, stochastic neighbor embeddings, are really, really, really useful actually, and very common in biomedical applications nowadays. And UMAP, and there's also, you know, you can get all kinds of lower dimensional spaces by feeding the data through a neural network. And we'll see a couple of those applications in NLP. And there's differences between these, right? And the dimensions that you'll end up with will end up having more or less separation between distinct subgroups, right, within your data set. So, and especially with TSNE and UMAP, you usually see a lot more distinct clusters than you do with, say, PCA. Okay, so let's try to build some intuition about PCA, since it's, you know, as the sort of one of the most common ones. We're not going to go into eigenvectors and eigenvalues, and we're not doing any decomposition of a covariance matrix. We're just looking at this dinosaur, okay, and trying to build some intuition around it. So what I want to do here is capture the most information I can about this dinosaur in lower dimensions. So the PCA algorithm will start rotating this dinosaur, and it'll find the view where if you take a picture of this 3D dinosaur, you capture the most information that you can capture about it. So taking a picture is our equivalent of going from 3D to 2D. So we're reducing in dimensions. And the most information is the variance that we're capturing, right, with this, with each of the views. And that's shown in this blue dashed line. So you want this blue dashed line to be as long as you can make it. So imagine, like, if you look at the dinosaur from the underside, you'll just see like some feet, maybe a little head peeking out, right? That's not, you don't get a lot of information. You don't know how tall it is. You don't know how big the tail is, maybe. I don't know. You don't know if it has spikes on its back, right? All these things. So let's start rotating the dinosaur. So, okay, yay, now we found a view that maximizes the variance that we can capture, right? So this blue dashed line is much, much longer. And this becomes our first principal component. So we have PC1. And now we fix this axis and we start rotating the dinosaur around this axis to find the next most informative axis. So the purple line, right? So now we want to make the purple line as long as we can. So, you know, we rotate the dinosaur around and ta-da, right? Like you have a view that captures as much information as you can in just 2D about this dinosaur. So here we see it captures the height, it captures the length, and these may also be variables in your dinosaur biometrics data set, right? So maybe you can check what variables are sort of correlated with these. They don't capture though, like we've lost information. We don't know how wide the dinosaur is. We don't know about spikes necessarily, and some information is gonna get left behind, right? Okay, so that's just some intuition, and here's an example that's not about dinosaurs. This is a plot of the first two principal components with data from the 1,000 Genomes Project. And the 1,000 Genomes Project did whole genome sequencing of about 2,500 people from 26 subpopulations around the world. And here, I think this is what Mustafa maybe was talking about earlier, right? The colors are based on metadata. So self-reported like race and ethnicity, right, of those individuals. So each dot is a person. The positions on the plot are based on the genomic data and then running PCA on that genomic data. And you see how the first principal component separates Asian, doesn't really separate Asian from European, from African. Right, so that ancestry is separated with the first principal component, but the second principal component, as you start, you know, moving on this axis, start separating Europeans out of that group, and so on. So in practice, we do, it's fairly standard practice in genetic epidemiology to use the first few principal components as covariates in a model that you build. So this way you control for ancestry. You don't really super care about the interpretation of those principal components, that those are not the coefficients that you care about the most, right? You're just trying to use them to control for the main effect that you're trying to estimate. Yes.
We have another question from Mustafa. Are there any iterative techniques to start with an initial value and give us the number of informative variables at the end?
Yeah, we'll talk about variable selection methods as we go. Yeah, yes, there are. The quick answer to that is yes, there are, and we'll see them. Good questions, Mustafa. You're anticipating everything that's, so tells me that this is a natural progression. So let's switch gears and talk about dimensionality reduction in the domain of natural language. So this is a paper from Mikolov and his group at Google, and this was a paradigm shifting paper published in 2013. It introduced what is now known as the Word2Vec model. So it's in Word2Vec embeddings. You may have heard that term. Don't worry if you haven't, maybe it's very NLP specific. They introduced a specific type of neural network. They call it the skipgram model. And it takes the corpus that has, say, a vocabulary of 10,000 words. And in this case, they output just 1,000 dimensional vectors. So we went from 10,000, right, a vocabulary of 10,000 to 1,000. It's not quite as simple as that, because the skipgram model considers some of the neighboring information. So our starting dimensions is not the 10,000 that I'm simplifying it to, but I think it makes the point. So let's now take those 1,000 dimensional vectors, right? So it's just a line of numbers that's 1,000 numbers long, and then run PCA on that. And then we plot the first two principal components, just like we did before. So this is the plot of the first two principal components on those 1,000 dimensional vectors. And honestly, this feels like magic, but it's math. In the 2D projection, right, you see that the model learned relationships between the concepts. So the capital of China is Beijing, the capital of Russia is Moscow, and the model has organized these concepts in a way that their relationships to each other are true to how a human might organize them, right? The country with their capital city. And the amazing thing is that they didn't tell the model. There was no supervision information, right? They didn't tell the model cities and capital and countries and their capital cities look like this, right? The model just discovered this by itself, just by how it is constructed. And just to add to how amazing this is, right? You can also do some vector arithmetic here and get results that make sense. So take the France vector, subtract the Paris vector, and add the Tokyo one. And France minus Paris plus Tokyo gives you Japan. And this works out, right? And if you want to sort of look at the vectors and do it with the parallelogram, you can see that it works out. There is an issue here, right? Take a few more examples of this that are not so sort of vanilla like countries and their capitals, and you start seeing how our biases end up encoded in the language that the models are trained on. So in the lower dimensional space, you may also find the relationship man is to computer programmer as woman is to homemaker, right? So all of these sort of biases end up represented, because all that this is doing is taking words that humans wrote, right, and building a model out from them. And debiasing the word embeddings is a very active area of research. So there's a lot of work going on to try and remove that bias. And there are also those who don't think that that's possible. That's again a separate conversation. Okay, so that word vector stuff is cool stuff, but how does it help you with EDA? So let's look at a corpus of clinical case reports, and the authors here trained a 300-dimensional word-to-vec model. So they have those embeddings, and they then run TSNE to reduce the dimensionality further and get a 2D plot. So I don't know how well you can see the labels here, but each of the colors is a different sort of organ or different system, and it has the heart cluster is purple, and you see it's closest to the lung cluster. Does that mean anything, right? That's where it becomes EDA, right? So you start making these observations about where things end up, and you try not to read too much into it, but also generate some hypotheses that you can test that will give you a better idea of what's going on, right, with this clinical case report data. Why is the heart cluster closest to the lung one? Are there concepts or words that are in common between those two types of case reports? What are those? What might those be? So yeah, so that's where it becomes EDA. All right, shifting gears to modeling, and with a few minutes left, I think it's good time. So now we have to make some decisions, right? We've built some intuition about, and then now we have to make decisions about what to feed to our actual model. So let's assume, right, that we have, for simplicity, just like a classification task is what we're working on. We want to get to a situation where we can get reliable estimates of the effects of each of these variables. So we want to thin out the number of dimensions. So you can take a subset of them, or you can use the reduced dimensions as inputs. Here are some pros and cons listed on the slide for one versus the other. The main one is interpretability. And this one is important enough to basically make many of the other reasons not matter in practice. That because if you need interpretability, you're constrained by that, right? So just some methods are not going to be very good for creating your inputs, if that's a requirement. Yeah, so I also have the word multicollinearity on here, which I haven't defined, and that's just, it's a problem in regression modeling where you have correlated variables in the models and their effects end up diluting each other. So like, to make it super silly, say you have height measured in inches or a height measured in meters, and that's like 100% correlated, right? It's the exact same information, but you have both of those in the model. That creates a problem because they're not adding information, relative to each other. So let's look at that first left column from the previous slide. So I think this was Mustafa's question too, right? So what are some feature selection methods? There are some papers that have more comprehensive reviews than what I have on the screen here. But in practice, commonly what you would use would be a bunch of filters. So we've already talked about dropping variables based on there's too much missing data for them, so let those go. Drop variables because they're too highly correlated with others. You also drop those. So those are examples of a couple of filters you can apply, and that's easy pickings. With regression models specifically, there's also stepwise selection approaches, but they have some caveats that make them better suited as exploratory techniques. So, but they're worth a mention. And of course, there's a number of ways to do what we call regularization. And these are ways to reduce model complexity. So the L1 penalty for regression, so also called lasso regression, it shrinks the coefficient estimates, and some of them can end up being 0. And if the coefficient ends up being 0, then you can drop that variable from the model. So you might have a solution design where you first, you have, I don't know, a data set with 10,000 variables. You may build a lasso regression that's going to drop, say, I don't know, 8,000 from that data set, right? It has shrunk those coefficients to 0. And then with that limited data set, you're now no longer restricted to regression models, right? You can take those variables and then do your subsequent modeling with another algorithm. So you're not restricted to one, right? You can use it as just a way to select variables. There's, for other types of models, so XGBoosts, you can also tune pseudo-regularization parameters. So we don't really have time to go into boosted trees, but there's like the depth of the tree and gamma. There's a number of parameters that you can tune to effectively regularize that model. Yeah, and I've already talked about combining those models. So this is just the last 2 slides here. So we are nearing the end if you have a hard stop. I wanted to give an example of a specific algorithm that is less sensitive to high dimensionality than most of the others. And that's support vector machines. So this was this was developed by Cortes and Vapnik, so from Bell Labs back in a long time ago, in relative terms. And the idea here is that you're trying to distinguish between the X's and the circles by finding a hyperplane that gives you the maximum separation between these two types of data points, right? So the largest difference between the X's and the circles defines a hyperplane that you use to then make decisions on. So the data points, right, the observations that lie closest to that hyperplane end up being the ones that influence it, right? So if you see, if I remove this X right here, then this X becomes the closest one. So then my hyperplane might shift. So those we call, the support vectors. They're the sort of important ones for that classification. And because the rest of the data points, you end up, they don't matter so much, right? It's automatically regularized. So you don't have regularization parameters or pseudo regularization parameters for SVMs, because picking the widest separation margin automatically regularizes the model. And this has been shown that SVMs do indeed behave exactly like other machine learning algorithms that use more specific types of regularization. Okay, last slide, but I know we're out of time. How?
And we have a question also.
Okay.
So maybe actually wrap it up, Athena, so
Yeah.
we can get a couple of questions maybe.
Okay. So just a 10-second version of this slide is, how do you know if you've handled the high dimensionality of your data appropriately? How do you know you did a good job? There's a number of diagnostic measures you can check. I'm listing them there. I'm also a huge fan of SHAP. It's a newish package for explainability, and it's based on game theoretic principles for calculating, the effects of various features and attributing feature attribution. So, but really, ultimately, what you want to do is check that your model generalizes to data sets other than what it was trained on. So you want to test it on out of sample data and see, is it doing well? Right? That's really the ultimate test. So what we're talking about here is largely overfitting, right? And avoiding building an overfit model. Right. All right, that's it. So
hey, thank you, Athena. Appreciate everything. So we have, we are almost a minute, 5 o'clock is right now, but I think we can take one or two very quick questions.
Sounds good.
Are there any questions?
So just a comment, like, really good presentation, very informative, interesting, and easy to follow. There was a lot of topics that I wasn't familiar with, but again, it was great.
I hope it was accessible. That was my goal. Yeah.
There are 2 questions in the chat. Also, one
I'm
from
happy
Mustafa.
to say I have a few minutes. Yeah.
So we have a question that says, are there any powerful unsupervised techniques for feature selection? So can you use basically a supervised techniques for feature selection?
Yeah, so when you usually say unsupervised techniques, most of those will end up suffering from the dimensionality too. Think of KNN, right? Like k-nearest neighbors. You end up with a lot of problems there just because of also how you calculate those distances in such a high dimensional space in a way that makes sense. So it's not how I would do it. If you know something that I don't, let me know.
Great. I think that that wraps it, actually, the last thing. So again, thank you, Athena, for the great presentation. And tell us a little bit about how you ended up in this space. And thank you all for coming today, and looking forward to seeing you in our next data science, Meet the Data Science seminar, which is when, Janina, can you remind us?
It's October, oh, sorry. 1 second, I don't have it right in front of my brain. October 20. October 20.
October 20. Okay, there's no other. Okay, great. Fantastic. Thank you all, and see you in October then.
Thanks for sticking around.
Thank you, Athena.
Bye.
Thank you, Athena. Have a good day.
Bye.
Bye.